I'm a biologist, and I work as a professor at the Institute of Biology at the University of Mexico. And I've been having collaborating with Professor Sahotra Sarkar from the University of Texas for many years now. So this will be a joint presentation. Before I start, I want to explain that as a biologist, we will be addressing emerging zoonotic diseases from a biological standpoint, and particularly studying the vectors and hosts that carry on pathogens and transmit those pathogens to humans. So in that sense, our interest is on putting these emerging zoonotic diseases of public health importance into a geographical context. So what is the problem? Here we have a young man with a severe mucocutaneous leishmaniasis, and which this photo was taken in Guatemala. And this is a disease that is a widespread disease, and actually, as I will show later on, it has been occurring lately in the United States as well. So it's a really huge problem, really high burden for public health authorities, and it has been regarded, this disease, as a poverty disease because it occurs mostly in rural areas in Latin America. So what is the challenge about studying these hosts and vectors of these zoonotic diseases of human health importance? Well, let me okay. The challenge is that we know very little about the reported cases in humans, as well as the host or reservoirs that carry on pathogens, and as well as the known vectors that transmit those pathogens into humans. So there's a huge area of research to try to make inferences on giving what we know about these reported cases and known reservoirs and vectors. How can we discover what are the unknown vectors and unknown hosts of these pathogens? So how can we understand and describe what's going on a huge area of research? So to address this challenge, or these challenges, I will describe these two objectives in my talk. And first of all, I will try to identify potential hosts, mainly small mammals and vectors, which are mostly insects, of two zoonotic diseases, which are Leishmaniasis and Chagas disease, which are really widespread, and millions of people are infected, mainly in the And to do this, I will use a statistical approach called data mining approach. And once we have identified these potential hosts, and vectors of both zoonotic diseases, I will produce potential risk areas under current and climate change scenarios using a very popular scientific tool called species distribution models. And I will refer that to ecological niche modeling approach. So I will describe those two objectives using these two zoonotic diseases as a case studies. So what is data mining? Well, data mining is a statistical tool, you have probably heard about that, that explores and analyzes data, relations, and other statistical associations. And I will use that into a geographical context. And you see here a map of Mexico, where I have overlaid all the localities where mammals have been collected throughout the country, and that is depicted in red dots. These data have been obtained from natural history museum collections worldwide that have collected mammals in Mexico into that are repository into huge databases. And I have also selected or overlaid the Lutzomia localities, which Lutzomia are the sandflies responsible for transmitting the pathogen that produces Leishmaniasis. And these green points are all the collecting localities occurring in Mexico of sandflies that are known. So I don't want to get very much into details, but basically the data mining approach examines the overlay between the red dots and the green dots into a null model or random model. And if there is a significant spatial association between the red dots and the green dots, then we calculate that as an epsilon value. And if this epsilon value is more than 2, it implies that there is a strong statistical significant positive correlation in a geographical context between this vector that is the sandflies, the green dots, and the host that is the mammals, which are depicted as the red dots. Now, here is a list of mammals, the first 2 quartiles of mammals, and here are depicted in these circles the 5 previously known hosts of mammals carry on the pathogen of Leishmaniasis, that is Leishmania. And this is the first quartile depicting the species with the highest epsilon values. And you can see that all the previously described hosts of mammals occur in the first quartile. So the probability of randomly finding 5 confirmed mammal reservoirs in the first column is about 10 to the 8, -8. So it appears that we have a good list of predictions of potential host of mammals that can be ranked as highest potential a host of mammals carry on these pathogens. And this can be depicted in networks of how the sandflies are associated with the species of mammals potential hosts. And you can see these red dots, which are the sandflies. And for example, this sandfly, Lutzomia longipalpis, is associated only with this group of mammals and shares some species of mammals with other Lutzomias, as you can see in this part of the network. And interesting, there are some parts of the networks where, for example, this sandfly species, Lutzomia anthropophora, does not share any of the mammals where they are distributed, occurring in Mexico. So this network you can derive a lot of information or make inferences about the geography of this zoonotic disease. Moreover, from a biological standpoint, you can see that from a parasite or pathogen point of view, if you are moving in this region of Mexico, you have a lot of interchange between the pathogen populations carry on from one sandfly to another and from one mammal species to another. But in this area, the gene flow appears to be more restricted, and in this area probably is more isolated. So this, from a biological standpoint, this opens the question if you are dealing only with one species of pathogen, or probably is a mosaic of species of pathogens. And Leishmania presents different kinds of diseases from Leishmaniasis. You have a cutaneous Leishmaniasis, but also you have a visceral Leishmaniasis. And so far, it has been only attributed to one pathogen, which is Leishmania mexicana or Leishmania brasiliensis or so forth. So this opens the question if there are the possibility of more species of pathogens, given the differences in potential gene flow between the pathogens. We did the same for Chagas disease, which that involves as well small mammals or mammals as hosts, and the kissing bugs, which are Triatomine species, the Triatoma species, and as well depicted here as in blue dots. And again, this Triatoma dimidiata is related geographically only to this group of small mammals. And this contrast with this part of the network where you have lots of triatomine species that share many small mammal species. So again, it has been only identified one pathogen, Trypanosoma cruzi, but again, it would be very interesting to see if we have a mosaic of species rather than one species affecting or producing different symptoms of Chagas disease or different types of virulences of Chagas disease in humans. So this is an open question that I think need to be addressed systematically. So once we had this potential host species, what we did was we conducted extensive field work in Mexico to capture insects. This is a daisy trap used to collect sandflies. And here we have Sherman traps, very typical to collect small mammals, particularly rodents and shrews. So we trap the rodents and carry them on into the lab to take the tissues. and conduct the PCRs to identify the pathogens that were present or not present. So one of our main findings was that we found some bats infected with Leishmaniasis, and this was a very important finding because bats fly long distances. So this allows the pathogen to disperse for long distances and probably infect other sandflies that are in other areas. This was a very important result, as well as you can see here a list of mammals that occurred in the first quartile in our previous graph, and all the collected mammals were infected at some degree with the pathogen of Leishmaniasis. So our list of potential hosts were confirmed, and so that this statistical approach appeared to be very robust in predicting new hosts of the pathogen of Leishmaniasis. So next, what we did was to use this list of host mammals and vectors that we identified on both diseases to produce species distribution models and in consequence potential risk areas under current and climate change scenario. So I will not go into details on how these species distribution models are built, but it's very popular. Many labs in the world are now using this methodological approach to project ecological niche models of species using museum specimens to produce potential distribution of species. And here we can see 4 maps of sandfly species that are incriminated as very efficient of transmitting this pathogen into humans. And you can see here the point localities obtained from museum collections, and the gray areas are the potential distributions of these species occurring in Mexico. And I just wanted to take a look at the species because these species have already been reported in the US carrying on the pathogens. So we have Lutzomia shannoni, Lutzomia cruciata, Lutzomia panamensis. And one of the very interesting results is if you overlap all these distribution maps into a single map, and you overlap the number of human cases reported for Leishmaniasis, virtually all of the human cases reported fall within our distributional predictions of the sandflies. And this makes a lot of sense, of course, because you have the vectors infected with the pathogen, you have the host infected with the pathogens, and then you have the humans living in these areas, and of course, the exotic cycle occurs. Now, this map also provides additional very interesting information, and these are, for example, these areas where there are no human cases reported, although it is predicted of a high risk, this area as well as this area. And there are a couple of possibilities that can be occurring in these areas. One is that Leishmaniasis occur in these areas, and human cases occur in these areas, but public health authorities are not reporting that for any reason. It's not clear which reason. The other possibility or explanation is that probably the sandflies, which might be infected with the pathogen, are not as effective of transmitting that pathogen as other sandfly species. So there might be a difference in the transmission ability of the vectors to transmit the pathogens to humans. So this needs also to be addressed in future research. But the interesting point that I want to raise or emphasize is that by following this theoretical and methodological approach, you can produce a risk map that appears to be highly predictable of the human cases occurring, at least of Leishmaniasis in Mexico. Now, these are the models, the distribution models of two sandflies that we produced some time ago, which are sandflies that are very effective of transmitting Lutzomia into humans. That is Lutzomia anthropophora and Lutzomia diabolica. And here are the climate change projections of distribution projections of both species under different under different future years. And you can see that the projections are cover wide areas of North America. Now, how reliable are these maps that we published in almost 13 years ago? Well, a recent study that was published in 2021 already reported some of these sandflies that occur in Mexico in some wide areas of the U.S., like Arizona, Oklahoma, and Texas, with the pathogen, which is Leishmania mexicana, Lutzomia cruciata, Lutzomia diabolica, and Lutzomia shannoni, in wide areas of southern U.S., even in some parts of the Midwest and even in the north, like Maryland or so. So the message here is that any species, biological species, respond to climate change, and climate change affects its distribution. So if these species happen to be vectors or hosts of pathogens, of course, they will disperse, carrying on the pathogens. And so we will find new areas of zoonotic diseases of human health importance where previously there was no reports. And this appeared to be the case in the US. So this is a composite map showing an overlay of all the triatomine species reported in North America, in Mexico, that is. So predominantly in Mexico, and the red areas depict the highest concentration of triatomine species. So we have lots of triatomine species in the west of Mexico, in the Baja Peninsula, and in the northern part of Mexico, as well in the northwest part of Mexico. But the projections also include some parts of the southern US. So I took this map from a publication that was in 2011, by Byrne et al., and showing the triatomine species diversity in the continental United States by county. What's interesting is that the states shaded gray have reported at least one species, and you can see really wide areas of the United States already being reported with triatomine species. And if you compare this map with my previous map, there is some geographical concordance between both maps. So it appears that these species distribution models can give really reliable hints or predictions in terms of where are these vector and host species carrying pathogens moving under current and climate change scenarios. So this is kissing bug, so that if you don't know or you're not familiar with kissing bugs, I will introduce this. And this Triatoma pallidipennis is mostly distributed in central Mexico and is one of the most efficient triatomine species transmitting the parasite into humans. So these risk areas can be really make fine-grained predictions, and this is an area located in central Mexico, and the red areas are where a high number of triatomine species occur, and the white dots are some of the human cases that have been reported in those areas. And if you take a look, even at these small scale maps, you can still have reliable predictions using this methodological approach.
Hi, Dr. Sánchez-Cordero. You just have 2 more minutes left.
Okay, so we did the same modeling exercise for 2 species of triatoma, the Triatoma gersteckeri, into the United States. As you can see, there are, under climate change scenario, there are predictions on wide areas of the United States, as well as these other species of Triatoma sanguisuga. And so this eventually will hold all the vectors, potential vectors and potential hosts, that if the pathogens arrives there for some reason, either by migrants or by animals dispersing to these areas, there might be some areas suitable for transmission of Chagas disease in the United States. So as a conclusion, I have 3 conclusions. The wide and millions of museum specimens that are held in natural history collections provide an important source of information for studies on zoonotic diseases of human health importance worldwide. I only shown these 2 cases, study cases, but you can use other zoonotic diseases as well. And data mining, this statistical approach, appears to be a very robust tool to identify potential vector and host species of emerging zoonotic diseases. And finally, species distribution models can help to identify risk areas of these zoonotic diseases under current and climate change scenarios worldwide. Muchas gracias.